Good morning, good afternoon. Welcome wherever you are. Welcome to the Global Prebiotic Association Virtual Education Series, where we're going to be talking about prebiotic mechanisms and immunity for the next several hours. I'm Len Monheit, Executive Director of the Global Prebiotic Association. It's really my pleasure to welcome you, and I think we've got a couple of really interesting hours ahead. The Global Prebiotic Association is now approximately 3 years old, a little bit over 3 years, and our mission is to champion the prebiotic category by increasing the awareness and understanding of the science, supporting both well-known and emerging benefits of prebiotics, and create needed transparency about prebiotics and their interaction within the microbiome as we learn more and more. Some housekeeping notes for the next couple hours. You will see on the bottom of your screen a chat button. You can send a message to the hosts and panelists, and our team will be triggering some interactive aspects and posting some notifications using that chat button. We also have a Q&A link at the bottom. You're really welcome to submit questions to our panelists. We'll do our best to answer them. I think we've got a lot of time, so we should be able to answer them live. But we'll also follow up on any others that we do not get a chance to post-event where possible and where needed. You'll notice that this presentation is being recorded. It will be available on demand early next week. You'll receive an email notification when the on-demand recordings are available. We've also got a resource portal on the GPA website where we'll have access to the slides and additional GPA prebiotic resources. Those also will be available early next week. And you can connect with the Prebiotic Association using our social handles. The scope of today's presentation is going to include a discussion of prebiotic mechanisms broadly, then dialing in specifically to mechanisms around immunity. We're then going to talk about prebiotic product development and communication and commercialization. And then we're going to finish up with a future of prebiotics interactive discussion that is open to all. So you're going to promote, it's going to be interactive. We're going to introduce ourselves and talk about the subjects that matter in today's world and tomorrow's world. Now, I need to say from a disclaimer standpoint that the information in these presentations is intended for educational and scientific purposes only. It's not intended as medical or nutritional advice for the treatment or prevention of diseases. For medical advice, please consult your healthcare professional. To start us off and to set the context for today's presentations, let's understand exactly what's happening in the world of our influences and our customers who are the ultimate consumers. So I'm going to talk about some consumer and dietitian insights as presented by the Ingredient Transparency Center, which does surveying of consumers as well as dietitians, has for a number of years in a broad range of categories, including prebiotics. So we're going to start with a consumer perspective. Consumer perceived benefits of prebiotics, and this is from the 2020 data, the 2021 are in process. And you can see that this is divided up into regular prebiotic users and those that are less regular. And you can see that predictably gut health and digestion is the strongest benefit in both categories, followed immediately by immunity health and then microbiome health. So immunity health is positively associated and strongly associated in consumers' minds with as a benefit of prebiotics. Now, from a dietitian perceived benefit, it's a little bit of a different story. You see that gut health and digestion is at the top. And actually, there's two separate groups of dietitian. There's a group of dietitian that prefers food first very strongly. And then there's a group of dietitian that are both mixed food and dietary supplements. So you see gut health and digestion at the top, followed by microbiome health to complement a probiotic. So sort of getting into that symbiotics. And then symbiotics comes in a little bit later. But actually, immunity health and autoimmune is a little bit down the list. And that actually it's the prefer food first dietitians that are much more aware of immunity health benefits. The purchasing decisions in consumers' hands on why they buy prebiotics. First of all, They look for, it's labeled as a prebiotic. They're looking for fiber. They're looking for a probiotic-prebiotic combination, and look for specific activity on the gut microbiota. So there's a diversity of influencers to the purchasing decisions in consumers' hands. From a dietitian standpoint, in their recommendations on why prebiotics should be purchased, you can see that the strongest response is looking for specific prebiotic ingredients. After that, it's looking that it's labeled as a prebiotic, and then looking for a symbiotic. So there's different communications pathways, different levels of awareness, and this is the landscape that we've got with both consumers, our ultimate consumers, as well as dietitians as a strong influencer set. So that's my tee up for the session. And now it's my pleasure to introduce the Global Prebiotic Association Science and Technical Director, Nathan Gray, who's going to lead us through, as workshop chair, the next couple hours of presentations and discussions. Welcome, Nathan.
Thank you so much, Len. Absolute pleasure to be here. Great insights there. And yeah, it's my job now to talk about, to introduce the rest of the speakers and host the rest of the workshop. We're going to start with a little poll though. We've got a couple of interactive polls coming up. So this is just a quick question to make sure you're all there and listening in. And this is which best describes your interest in prebiotics? Are you watching the space closely? Maybe just got a toe in the water? You're a strong believer and committed? Absolutely all in. It's good to see where everyone is in this, and most we've got to just going to give it another couple of seconds. Okay, we're sharing the results now. So as we can see, a great chunk of people are either strong believers and very committed to prebiotics, or all in, but just over a quarter watching the space as well. So welcome to those who are watching the space. Hopefully this is going to provide some great insights and actionable things for you. Sorry, I was just trying to clear my screen there. So as Len already suggested, we've got a great couple of hours ahead. This first session is looking at prebiotic mechanisms and immunity in focus. We've got 3 great speakers plus myself to go through over the next 90 minutes or so. I'm not going to waste any more time in introducing everyone. I'm going to hand over first to Stephen Fleming, PhD, co-founder and president of Traverse Science. And he's going to be here to present to us on some of the mechanisms on prebiotics and immunity and general prebiotic mechanisms. Welcome, Stephen.
Excellent. Super happy to be here.
Thank you so much.
Ready to go? Ready to do this?
Great. If you're able to share your screen, I will hand over to you right now.
I sure am. All right, let's do, we're going to pick a risky move, share the whole screen, not just PowerPoint. This is great to be here. I picked all in on that poll, and I unintentionally ended up doing a PhD in prebiotics. I was supposed to do it on choline, and that didn't end up working out. Something about a freezer going down with all of our samples. But when I first started working in this area, I was like, who cares about prebiotics? These are just things that help you poop better. I was in a neuroscience program, so of course that was where I was, but I was very pleasantly wrong about that. And I'll be talking about some prebiotic mechanisms and immunity. My background is in neuroscience, so it might be like, why is this guy talking to us about immunity? But I've been around a lot of projects with an immune focus, and I think a lot of the lessons learned cross over really well. So let's get into this. If it will, let me pull up PowerPoint here, actually. Okay. Okay, I swear I know how to work PowerPoint. All right, definitions. I don't want to spend too much time on this, but all of the biotics, you can see what I'm trying to say here, have some commonality between them in that they have to confer a health benefit on the host. Whether we're talking about poultry, pigs, pets, humans, the health benefit might be different between them, but this is the commonality between these that I'm usually most interested in. I'm mostly on this side. And I used to do a lot more work in mechanisms. Now I'm in a position where I'm a service provider and a consultant, primarily working with ingredient suppliers and consumer packaged goods companies. Probably the most requested type of project that we work on is claim substantiation. And, there's some trickiness to the biotics because some of these are new categories. Regulatory agencies don't always have the best guidance out for if something actually is or isn't one. We're obviously talking about prebiotics today. Here's the GPA definition: a nutritional product and or ingredient selectively utilized in the microbiome, producing health benefits. Of course, there's important mechanisms in the selectively utilized part. I think speakers after me will be really good at that. And so you don't actually need a mechanism to produce a health benefit. So, do we even need to talk about those? I still do think it helps. So what I'm going to talk about is really, where do mechanisms fit in the broader picture? I'm speaking more from the perspective of towards an ingredient supplier and a CPG. But I think it's also helpful if you're an academic researcher, or maybe you communicate in this space. And if you'll allow me to do a little bit of marketing corporate speak for a second. I'm almost tired of hearing stuff like storytelling. But this is the hero's journey. I'm rebranding it to call it the prebiotic journey, where you kind of go through this cyclical process, right? And we're all the heroes of our own stories, right? Whether we're creating a prebiotic, incorporating it into our ingredient, trying to promote prebiotics for health and wellness. So maybe our call to adventure here is prebiotics. And through some supernatural aid, like an R01 grant, maybe venture capital funding, maybe like a health claim or something like that. Right? Maybe those are the things we're trying to get to. And mechanisms, I think, will be helpful in doing that. And, at the most basic level, right, it's going through this science and trying to understand, what do we know? What is the current state? There's so much opportunity in this space, especially on the clinical side. If you can see my video, I worked up here in pigs, and there's so much great and interesting preclinical research. Then you start doing those reviews and you're like, what do we know clinically? And, those search hits start really narrowing down. But then, you go through this process of, all right, let's do some in vitro trials, maybe get some fecal inocula, do some fermentations, graduate to small animal, large animal trials, maybe clinical trials. I think, especially if you're in a business or if you're doing academic work too, we want to just avoid this part of the hero's journey entirely. Let's have no abyss, no deaths, no rebirth. But if it does happen, maybe your clinical trial shows there isn't a health benefit, like the worst possible probably outcome there. You start saying, well, how are we going to pivot around this? But ultimately, I'm just showing this because there we go. I think you can take a pessimistic and an optimistic view to mechanisms, right? I think if you're able to show your audience, other scientists, healthcare professionals, consumers, maybe you're trying to sell your prebiotic to a CPG, if you can point to the mechanisms along the way that say, well, okay, here's the structure, it's fermented by XYZ bacteria to produce these pathways, and you can draw these nice clear lines, which is very difficult to do. Even if you can only do a few of those things, I think it really helps to build trust and understanding with your audience. And, your audience may not really be as into the science as you are. So it's really important to be able to build that trust, and mechanisms can play a big role in that. Even if you're not on, say, a communication side of this, understanding mechanisms will certainly help drive R&D and help with the creation of derivatives. Early on, I didn't realize GOS and FOS could take multiple forms, right? There could be different chain lengths of those. They could have different residues, different branching in a lot of these. Some of them are derived from fibers, and the structure of that fiber informs the structure of that prebiotic too. And so that's where maybe sometimes you kind of pivot around the structure of it. Of course, the pessimistic view is, look, just to be honest, it's really expensive and time consuming to look at mechanisms. We're here in 2021. We've known about a lot of these prebiotics for a long time. Sometimes I feel like it's just as much of a mystery today as it was several years ago for how some of them produce the effects that we think they do. It can be confusing. And of course, with all science, this isn't unique to prebiotics. You finish a study and you just have more questions than you have answers. So where are we in the journey? This is actually a figure stolen from the postbiotic paper that ISAP put out. In my opinion, we're not very far. And I say that in a in an optimistic way, to say that there is so much opportunity to learn more about how prebiotics work and what are the mechanisms. I think these are the really common ones that, they're almost de facto discussed in discussions and introduction sections and papers, right? We think, all right, well, we're going to feed prebiotics. They may modulate the resident microbiota. Pathogenic ones go down, beneficial ones go up. They produce fermentative metabolites. And, we know from preclinical evidence and in vitro that those metabolites should affect barrier function. And it's not necessarily that 1, 2, 3, 4, and 5, these things all happen in a linear manner. But, if you're to tell me that taking a prebiotic, for example, reduces susceptibility to infectious disease, whether that's, bacteria or a virus, or in the case of my research, what was getting really popular, and I think still is prebiotics and things like anxiety and depression. There's a big distance, I guess, physically, but also scientifically, between saying fermenting selective utilization of these things produces an effect like reduced depression. And it's going to take a lot of convincing for me to do that. Now, back to the health benefit aspect, I'll use an example from the FDA. The FDA has some nice guidance on what constitutes a dietary fiber, and EFSA has some opinions on this as well. Short chains, fermentative metabolites, and microbiota, those are not health benefits. I think a lot of us active in this space want to see them be treated like true biomarkers. But short chains can go up, go down, go sideways, and have no effect on your ultimate health benefit or have no relationship to them. And the same can be true of the microbiome. I'm scooping myself here, but the point I think I want to make throughout this is that context is key. And when we look into mechanisms, I think some of us, we do just want to have a basic understanding of how this works. I think many of us are interested in, well, is there a biomarker of some process that I can pull out? If you're counting B cells and T cells, can you maybe predict the population of those cells by maybe one of these other measures? Sometimes some of these mechanisms are hard to capture in clinical populations. So if we could maybe have a better idea of how the microbiome and metabolites in a fecal sample relate to these other mechanisms, that could help us build that mechanistic story for here's why we see a health benefit. And what I'm saying is that mechanisms are contextual. You can change the microbiome and have the same functional output, and it can appear to have the same genera or species or strain in there, and then also have the same functional output, right? So I don't think all of these steps are necessary. And of course, these are just examples. And I'm going to give a more concrete example there of what I mean by this. Well, I'll do it right now, actually. So when I say initially I worked on prebiotics unintentionally, they were on HMOs. What's an HMO? Okay, human milk oligosaccharides, and there's tons of different types of them. And we were working with Nestle and feeding these to pigs, different types of them, trying to understand how they affect brain development. And we were finding this weird effect where some of them affected cognition, some didn't. We weren't able to figure out if it was a dose or form thing, but they would even affect short-term memory and long-term memory differently. And so I'm pulling up just some select figures from this paper. We're not going to go super in depth, but if I come over here on the left, we fed pigs no oligosaccharides, bovine milk oligosaccharides, which were mostly galacto-oligosaccharide and some sialactose versions, human milk oligosaccharides, which were 2-fucosyl-lactose, lactoin, neotetraose, oligofructose, and phos, or adding 2FL. All I'm showing here, it's the phylum level, very broad, bird's eye view, looking at genera in the ascending colon and fecal samples. Honestly, very similar between groups. This is showing individual animals. It's not showing you, box plots and ANOVAs and things like that. But the microbiome is really similar. Even surprisingly between ascending colon and fecal samples, which we know are very different in metabolites, at least, a fairly similar population. If we go through and look at what we called common genera, those that were abundant at greater than 1%, and just looked at median relevance and make some of these heat maps, Sure, yes, you can kind of pick out like a few, you know, genera that seem like they're different, but broadly speaking, they're pretty similar. If we look at what we called uncommon genera, relative abundance of 0.05 to 1%. Again, you know, you might say, oh, Prevotella, something might be going on in this group. But overall, when you add the stats to it, they're not altogether too different. You know, and we did the whole let's do PCOA plots and linear discriminant analysis and try to see how they separate. These animals didn't really separate much on the microbiome, which, you know, makes you think like, well, did this trial even work? You're feeding HMOs. There's all this evidence from other work that they're selectively utilized. Why didn't the microbiome change that much? So whether you agree that these things are prebiotics or not, they're definitely oligosaccharides. Our take home was that these prebiotics had little impact on relative abundance of genera. I'm definitely aware we should really be looking at species and strain level, but, you know, we have the data that we had. So the question is, I guess what I've alluded to earlier, can the microbiome be the same and you still have some change? And what we ended up doing is taking a mediation and moderation approach. And if you're not super statistics savvy, this is the example I like to give. And let's say you want to know if the microbiome effect or is related to some distant variable. Let's take this example of a stove. You know that if you turn that stove on and you change the dial of the knob, that is eventually going to increase water temperature. You know, if you're trying to boil a pot of water, for example. I mean, we know that like the knob itself isn't magically making the water temperature go up. It is turning on the flame, or if you're in my house, the electric stovetop. And it's that heat source that is then truly affecting the water temperature. So we would say that the flame here, or the heat source, mediates the relationship between the knob and the water temperature. You know, although there's a very, very strong correlation between the knob and the temperature of water in that pot, we know that that's not a causal relationship. Causal would be more like this flame. And the reason it's not causal is because, well, what if you turn the oven off or you just like unplugged it? You would see that there is no relationship between any of these things. I mean, they would all just flatline, right? And so I would say that the flame is a mediator. Mediates the relationship between two different things, something that is a predictor and something you're trying to predict, the predicted. The power moderates those relationships. These relationships exist. This just moderates how strong that is. So let's replace this oven example with a biological one and say, what if we want to know how the host microbiome, measured through 16S sequencing, affects cognition, measured through this thing called the recognition index, which measures memory. As I said earlier, you know, why would a different population affect cognition? There's really not a good answer. It's got to be acting through some other pathway. So maybe the theory here is that, well, changing the host microbiome, probably through many mechanisms, ultimately has an effect on brain development, measured by hippocampal gene expression. And that itself is a much closer predictor of memory and cognition. So gene expression here might be the mediator, the way that the microbiome is acting upon the brain, but that might be mediated by intake of oligosaccharides of different prebiotics. So we looked at, actually, I think we looked at 6 diets in that study. We looked at 5. I mean, do the math, 5 times 3 different pathways. There's like 15 places to look. The diet introduces so much complexity, which I'm sure many people attending this can appreciate. And I'm just going to show a little slice of this. You can go check out the full paper, but if we break this down and we look at just pigs that we had fed oligofructose and 2-fucosyl-lactose, we looked at short-term memory for starters and found, hey, there's some genera that are positively correlated. Again, correlation doesn't equal causation. And I've colored these lines to show the direction of that relationship. So blue meaning positive, red meaning negative or inverse. So you'll see that all of these genera are positively related to short-term memory, meaning higher relative abundance meant better memory. And if you look at gene expression, well, those all have red lines attached to that. So lower expression of these genes, if you're not familiar with them, we have a serotonergic, a cholinergic, GABAergic, and glutamatergic gene here. Covers quite the spectrum there. Lower expression of those was correlated with increased performance on memory. So think of this like a negative times a negative makes a positive, which is exactly how that works. So genera were negatively related to gene expression. More of these meant less gene expression. Less gene expression meant better memory. And when I say a biomarker is contextual, you might say, hey, these are biomarkers of short-term memory. Maybe you don't even need to measure memory, your health benefit. You could measure these intermediate things. The problem is when we looked at long-term memory, which if you're not in cognition type work, you might just hear memory and say, aren't they pretty similar? Neuroscientists would disagree. There wasn't even a graph for me to show you. There were no genera that were and mediators that I could make a model out of. They didn't have that relationship. And I'm only showing you one diet. If we looked at pigs that were fed an HMO or a BMO, this whole graph looked completely different. There were different genera, different mediators, with different relationships to the health benefit. And again, I'm kind of giving these examples of when, you can take a optimistic or pessimistic view. I think the simplest way here, we want to be able to say, hey, increasing Prevotella based on this, that seems like a good thing. Let's do that. So if we can run some in vitro trials and just increase Prevotella, that's great. The problem is it was so contextual to the health benefit and to what those animals were fed that it doesn't generalize very well. So I think to take this out of that specific example, just in that publication, in neuroscience, we have these concepts of divergence and convergence. A single neuron could diverge and innervate a bunch of other cells. And likewise, a bunch of cells could converge onto a single neuron. And I think we can take the same approach with biochemical pathways and mechanisms, where let's say we start with a prebiotic, that's our substrate. And maybe these first few steps in this mechanism is the selective utilization of it. Maybe these are different bacterial populations, these nodes. And when you're going through trying to understand how these things work, you may, end up in dead ends, which I'm representing as these red nodes, where you can't really go past that mechanism. Maybe the previous mechanism doesn't really relate to the next one. And on the end here, we might have multiple mechanisms that kind of converge to show that health benefit, right? Maybe that health benefit is weight loss. Maybe it's resistance to infectious disease. Maybe it's memory. And so you can look at both ends of the spectrum here. And I think what I'm trying to say here is the stuff in the middle, those are not health benefits. And sometimes they don't, and sometimes mechanisms that seem close are not actually related to the ultimate health benefit. And this whole graph can kind of change. And so if I put this in context of what I was saying earlier, in terms of these things being a journey, I think that this divergence and convergence here, this is why public-private partnerships are so great, because the middle represents opportunity for academics. This is where the interesting stuff is. It's the cutting edge, how all these pathways relate to each other. I'm not trying to say that, I guess I am saying the ends are boring here, which is just me being hyperbolic. We know that many prebiotics are selectively utilized. It may not be as interesting to show that over and over again. And we know that they produce certain health benefits. Being the next person to show they do that benefit, there's still opportunity there, but it's not as novel. Journals are like, okay, we've been there, done that. Let's see something more interesting. Whereas for businesses, the middle is risk, right? I mean, why would you plan a clinical trial looking at a bunch of this stuff and sink hundreds of thousands or $1 million into those without knowing what you're going to get? And the ends represent reward, because the ends of this is where you can really fit into this definition of being selectively utilized and producing or conferring a health benefit. So hopefully we don't get caught too much in this little abyss, death and rebirth. And so that's kind of where I want to end it. I know that wasn't a ton on immunity, but I think speakers after me are gonna be able to talk about that. And I'll be sticking around later for the panel and for questions. And I hope you enjoyed that. If you have any questions, feel free to send them along to engage@traversescience.com.
Thank you so much, Stephen.
Excellent.
Absolute pleasure to have you with us. Really fascinating presentation there. Hopefully everyone enjoyed it. If you do have questions for Stephen, he will be joining us for a group Q&A at the end of all of the presentations in this first section. So please do use the Q&A box down at the bottom of your screens. Do type away there. We're not just ignoring them. We will get to them in that group Q&A. For now, though, just a big thank you to Stephen for setting the scene. And I'd really like to welcome our next speaker, Pieter van den Abbeele, who's going to be presenting some really interesting data and insights on in vitro work on mechanisms and some other things. I'm going to pass over to Pieter to have him explain a little bit more about his presentation on gut models and in vitro work on prebiotic mechanisms. Thanks, Pieter.
Right, super. Thanks for the introduction. So, yeah, indeed, today I will talk a bit more about the value of in vitro models to really understand how prebiotics are impacting the microbiome and the other way around. So my background is that, like the previous speaker, I also did my PhD about prebiotics, but the focus was really to test them in vitro models and always also try to improve in vitro gut models to make them more realistic for the in vivo situation. So after 7-8 years at university, I spent also 7 years in a CRO that commercialized an in vitro model. Was there also the CSO the past years. And then in the course of last year, I took the decision to quit my job and start a new adventure in the same area, because I really felt that I needed to do something more than I was doing at this moment. And this led to the foundation of Prebiotics. And today's presentation will focus on the limitations that I mostly observed and how I am addressing this at the moment. And I hope by the end of the presentation, you agree that I was not a fool to quit my previous job, but that it made some sense. So first of all, of course, as you all know, prebiotics, they have a direct impact on the gut microbiome. So here's just an example from a study that I performed with humanized rats during my PhD. So in this case, for example, you see on the right control samples, on the left inulin-treated and arabinoxaline-treated rats-derived samples. And you see, of course, the known effect of a prebiotic is that they stimulate bifidobacteria, bitrate producers, and this is kind of the direct effect when you deliver a substrate, that these beneficial bacteria grow on the substrate, and then the metabolites indeed can have downstream effects. Now, also something that I really want to highlight, and what I also observed very strongly in this in vivo trial, was the fact that prebiotics have also a very important indirect effect, meaning that they change the environment of the intestinal tract by, for example, decreasing the pH. And doing so, they really determine also which bacteria can grow at these specific pH conditions. So for example, I observed in this trial also that the strong fermentation of prebiotics decreased also colonization of opportunistic pathogens, but also, for example, Akkermansia, which is then, but I also found out later, one of the most pH-sensitive microbes. It's been correlated to a lot of studies, but yeah, it's really very dependent on the environment. And it's the current in vitro models, they really focus on direct prebiotic effects, often by controlling the environment. And it's also the second part that I see as one of the limitations of current in vitro models, that I also wanted to do something with. Limitations that I noticed in some of the conventional in vitro macro models, how I call them here, that you see on the right, that have been really valuable for fundamental industrial research, but they're already there for 30 years. And what I see as their main limitations, that they're very complex to operate, labor intensive, resulting in error proneness of these models being very expensive, not being able to test a lot of conditions. And another really important aspect is that when you deliver a sample from the in vivo situation to a lab model and you operate the lab model under certain conditions, you always see a deviation between the sample derived from the human being and then the microbiota that is in the end growing in the reactor system. And this really is also questioning also the biorelevance of the simulations, also the link between the individual donor of an experiment and the results becomes also more vague. And as a result, the main problem that I noticed was when we are setting up experiments in this field, and we really rely also on in vitro models because it's very difficult to get in vivo samples from the site of location. The compromises that we have to make always is compromise between statistical robustness, looking at multiple donors, multiple individuals, because we know that there's a lot of inter-individual variability. and also being able to test multiple test products. So it's really always trying to make the best decision. And this led to development of a new technology within cryptobiotics, and we call this the Cypher technology. And this focuses on the gut microbiome simulations. So it's a semi-automated intestinal fermentation research. This is what the abbreviation stands for. And then sometimes, of course, you also need to couple colonic simulation to a simulation of the upper GIT, because not all fibers enter as such in the colon, but they might need in pre-treatment. The technology that we are working on is based on three very important concepts. One is miniaturization, because when you're developing new potential active ingredients, you don't always have a large quantity available. But also importantly, and that brings me to the second and very important point, is physiological relevance. When you're working with a smaller volume, for example, if we were talking about the upper GIT, you need enzymes. The current standard is using porcine enzymes, but when you work with smaller quantities, you can also use human-derived enzymes, for example, in some cases. So lowering the volume can also allow you to increase the relevance of the simulation. And then finally, that was what the co-founder of the company is really specialized in, is automation of lab operations, but also of data analysis and reporting. And before we go to the scientific part, just also to make it visual, how we work is we are a CRO, so we start from high quality fecal samples. What I mean with high quality, I will explain a bit later. We do ex vivo incubations, and then also, everything is geared for high throughput. So the fundamental fermentation parameters we measure in-house, and then advanced analysis we do it with partners. And like that, we really focus on making the incubations as representative as possible. And then, it really depends whether you're in early stage, whether you want to do 16S, for example, as a more screening approach, or when you're in the later stage, you do shotgun sequencing, and you go to more in-depth analysis that we then integrate in one report. So with that, I want to show you some evidence of the things that I've been mentioning before. And one of the key aspects that I want to start with is the biorelevance of the gut microbiome simulations in vitro models. So I was mentioning before that samples from an in vivo host evolve within a lab system. Now, for me also, I'm a bioengineer from training. I also wanted to have some quality control parameter, let's say, to really make this not just a subjective statement, but really something that you can objectively look at. So to quantify this bias, in vitro bias, what I did was apply two approaches. So one is an X-Y plot, like you see here, where on the Y-axis you see the abundance of a microbe in vivo in the human sample. So the higher the species, microbial species is positioned, the more abundant in vivo. And then on the X-axis, this is really showing the bias. This is showing the ratio between the in vitro abundance and, on the other hand, in vivo abundance. So then we take the log value. So if anything is positioned to the right, it means that the microbe likes to grow much better in the lab than it does in real life. When it's more positioned to the left, it's actually growing worse in the lab. So it's depleted in the lab situation. And to also being able to put one number on this kind of plot, what we also do is for the 50 most abundant species, we just make the sum of all these X values, so the difference between the Y-axis, and this gives you one score. So for example, for like a technology with a relatively small bias, so with all the bacteria being quite closely clustered to the Y-axis, you see, for example, here a score of 10. For a technology where you see that the microbes, they really deviate a lot from the Y-axis. This means a lot of them are growing better in the lab, a lot of them growing worse in the lab. Here you see a bias score of 50, for example. So until now, these are all more theoretical examples. What I then, of course, started from was to look at the examples from literature from the two most widely used in vitro models over the past decades. And what you see here in the microbial communities of these models, when you give them prebiotics, according to published data, this is the kind of plot that you get. So for the two models, I won't name them by name, but you see my point is very clear. So a lot of the dots are being positioned towards the right or to the left. So for example, here you see in the first model an example of a dot with 4 log units deviation. So this bacteria is actually almost absent in vivo, but it's increasing 10,000 fold in the lab system. So it's E. coli genus, for example. It's a typical issue that can happen. And you see both models have a score around 40. So what we then, of course, did within Prebiotics when we developed our new model, we developed an integrated approach going from collecting the in vitro sample, the in vivo sample, up to running the experiment under optimized conditions. In our first model, where we were staying quite close to the state of the art in terms of media, we were able to get a situation like you see here, with already much less bias as compared to conventional vitro models. And then upon developing over the past half year also new media and also new inoculation strategies, you see that doing this extra internal R&D, we really arrived to a situation where my samples, if you plot them, the microbial species, because this is based on shotgun sequencing data, they are not so much deviating anymore from the y-axis, meaning that you are really able to preserve the in vivo sample as quite optimally in the lab. And when you then give the prebiotic, the response is also going to be much more representative for the in vivo situation. So this was really a very important point. And then here, with this, I also quickly show some practical examples, like for example, for inulin, we did a test where we compared to the blank. And then when you look to metabolic activity, you see that, of course, when you do this, you work with this in vitro model, it's really reproducible. So you nicely can distinguish blank samples versus the inulin treated samples. And you see also very rapid fermentation within the first time point, on third time points also. So if you look to the underlying variation, as already quite well known and mentioned also before, that what inulin does, of course, is stimulating acetate, propionate, butyrate production. You see it also here nicely clustered to the right. So with this kind of in vitro models, you really are able to understand well the impact on microbial activity. Then we also did with Cosmos ID some shotgun sequencing on the same samples. And then here also you see the impact in the PCA of inulin on the right side, the blank on the left side, on the various time points, also showing that you can really nicely distinguish with these kind of in vitro models what is the true impact of a substance. And then what I also wanted to highlight is the underlying changes in this case family level. That's the families that's closer to the right were closely related to inulin. Those were some families that are really known to be directly impacted by treatment. But interestingly also, there were some groups, and I mentioned it, if you remember in the very first slide, that were indirectly impacted by prebiotic treatments like Akkermansia, Enterobacteriaceae. They were also nicely clustered to clustering with a blank, because of the fact that we also really optimize the technology to look at indirect prebiotic effects to be as representative as possible. Also, an important step was, of course, to see what we get when we compare it with high quality data. Can we do some good predictive work? And you see here, when we looked for inulin fermentation, in terms of total short-chain fatty acids, we were really quite well able to predict the total amount. There was some bias in the individual fatty acids, but overall, we are working in the same order of magnitude. Then also to add some more biorelevance to these models, what I did during my PhD was developing models not just for suspensions, but also for bacteria adhering to the mucus layer. And this is also what we have implemented in the Cypher technology. And I will go quite quickly through it, but what you see here, for example, is 4 samples from a suspension and then 2 from the mucus. And when we look at Lachnostreaceae, so bacteria known also in vivo to grow very well close to the epithelium, where they can really impact host health much more. We also see that in the in vitro model. And when we highlight here just a few species belonging to Rosburgia, you see that in the 4 lumen samples, they're actually absent almost. In the mucus samples, they are abundant up to 20, 30, 40%. So it's really showing that this in vitro model can also mimic that. And when we look at the inulin treatment effect, we saw nice increases in 2 strains. One of them, Rosburgia inulivorans, which is of course very relevant when you test inulin, to see as a nice increase. So again, showing by adding this extra complexity that it really makes the model much more biorelevant. Then a final example that I also wanted to show was in terms of the quality of the inoculum. It was already mentioned before by the previous speaker that between ascending colon and fecal samples, there is actually not much of a difference. So the fecal sample, meanwhile, we know that it's the representative sample to inoculate incubations. But the problem that we also noted is that in some models, The standard is to pool samples to a single individual to circumvent the limited throughput of such models. But many key opinion leaders also say that, yeah, it's not a good practice to do that. So we set up an experiment to also gain some information regarding this topic, where we tested for 6 different age groups, 6 different donors versus their pooled sample. For the sake of time, I will of course just give one single example here. And this is an example for 6 breastfed infants, 3 months old. The way how they fermented 2FL was that they of course stimulated 2FL stimulates microbial activity, some bifidobacterial effect, of course. And what you see here is the response for the 6 individual donors. Now, when you calculate the mathematical average, this is of course nicely in the middle. But then the question is, where would the pooled sample be? And when I now show where the pooled sample is, you see that it's completely out of the spectrum of an individual response. So what we actually do by pooling samples is creating a super inoculum with the best bugs from the various donors. And this is not really representative for what the individual donor was capable of doing. So you have a very optimistic view, let's say, of what a substrate can do. And it's not representative for the individual response. So here you see some other examples, but in the sake of time, I will not go into too much depth, but you see that with the new technology and the high throughput, you can gain also more understanding about much more different donors, in this case, donor categories in terms of age. So with that, I just want to conclude that what I'm really now trying to do is really push boundaries in terms of the throughput by relevant and also robustness of experiments, so that when you want to publish a scientific paper, that you don't always have to reply, sorry, it was not possible to test more than one time this experiment because of the cost that it would involve. And really try to avoid that we have to make this kind of compromises. So, and then as a final slide, what we really have been trying to do within Cryptobiotics is really make sure that R&D budgets are not spent to very complex setups, and you actually have to pay for the complexity of the experiment and not really for the output, but really try to understand mechanisms much better by being able to shift budgets from the complexity of the test to actually the generation of data. And within the same budget, you generate much more information, much more things that can really help you understand how a prebiotic is able to operate. And this is what we do at Cryptobiotics. So microbiome studies, upper GIT studies, custom projects, and I'm also actually working as a scientific writer and consultant to streamline in vitro data to subsequent in vivo trials. So I'm happy to help if somebody would ever need my assistance. So with that, I would like to thank you for the time that you listened to me, and we can have some questions later on.
Thank you so much, Pieter. Really fascinating presentation. I'm sure lots of people have questions. We can see a few coming in. Just a reminder that we do have a Q&A box down at the bottom there on your screen. Please open that up, type in your questions. We will get to them in that group Q&A session to come at the end of this session. For now, just a huge thank you again to Pieter. A great presentation, really, really insightful. So excited. I've got a lot of questions for you. We're just going to head along with the next poll question now. So in which of these areas do you think the science on mechanisms is the strongest? So digestive health, immune, inflammation, or it's not strong at all? And as I say, if you do have any questions, please do pop them in that Q&A section. Please do stick around for the group Q&A towards the end of this, and also the second session, which will also follow the same format. So here we go. Here's the screen sharing for the results. We can see digestive health wins out by an absolute landslide there. Lots of people of the opinion that digestive health really is where the majority of mechanisms evidence for lies. I completely agree. 17% on immune, 10% on inflammatory. Thankfully, only 7% saying it's not strong at all. Although that is still, a good couple of people that are showing, suggesting that it's not strong. Hopefully, we can start to convince you otherwise. For now, though, I'd like to move on to our next speaker. I'd like to welcome to the virtual stage Stefania del Fabbro. She's a PhD student at the University of Southampton. She's been working on all sorts of wonderful things, but she's here to talk to us today about prebiotics and their non-microbiome related mechanisms. That's correct, Stefania, yes. Welcome.
Hi, everyone.
We can't hear you very well, Stefania. There you go, we can hear you better now. There we go.
Can you hear me? Yes.
It's still a little bit muted. I don't know if you...
I should be unmuted now.
There you go, it's better,
Yeah.
that's better. Yeah,
Super.
okay. So I'm gonna head off now and hand over to you. Thank you so much.
Thank you so much for the invitation. So as you anticipated, I'm a final year PhD student at the University of Southampton, working in nutrition and immunology under the supervision of Dr. Karen Childs and Professor Philip Calder. And today my talk is going to be about the direct effects of prebiotics on immunity, which are so far less explored than their microbiota mediated effects. So I'm going to start with an overview on non-digestible oligosaccharides and their prebiotic mechanism, to then move on to their microbiota independent effects, and then highlight the potential beneficial role of non-digestible oligosaccharides in inflammatory bowel disease, and finish off by mentioning mucosal associated invariant T cells or MATE cells, which have a key role in gut immune interactions. So what are non-digestible oligosaccharides? So those are dietary fibers that are resistant to the acidity of the stomach and reach the intestine virtually intact, where they are selectively fermented by health-promoting bacteria, for example, bifidobacteria or lactobacilli. And these bacteria break down the fibers and then use them as an energy source. And in turn, they produce short-chain fatty acid as a volatile byproduct. And those short-chain fatty acids have known immunomodulatory properties, and examples of those are butyrate, acetate, and propionate. And for example, butyrate is known to increase the amount of regulatory T cells, which are involved in controlling immune responses. So here I'm showing you some structures of commonly known prebiotics non-digestible oligosaccharides, and the most well-known are inulin, fructooligosaccharides, galactooligosaccharides, and the emerging mannooligosaccharides and xylooligosaccharides. So traditionally, the effects of prebiotics on immunity were thought to be related only to the modulation of the microbiota or the activity of their metabolites, such as the short-chain fatty acids or other antibacterial compounds. And examples of effects on immunity include the growth of supporting the growth of bacteria with immunomodulatory activity, the inhibition of pathogens via different mechanisms, including competition for nutrients, inhibition of adhesion to gut cells, and the production of antimicrobial compounds. In addition to this, also the secretion of short-chain fatty acids, which affect cytokine expression and the functions of immune cells, the maintenance of epithelial barrier integrity, and finally, they might enhance the antibody response to seasonal influenza in older adults and prevent allergy and infection in the first 2 years of life. However, recently there is a growing interest in understanding whether prebiotics may also modulate immunity in a microbiota-independent manner, especially in those individuals that have increased gut permeability, for example, in infants which have an immature gut, or in those conditions such as IBD, or which are inflammatory bowel diseases, or obesity, type 1 diabetes, or non-alcoholic fatty liver disease, which are all related to disruption to the gut barrier. And in addition to this, there might be also cases where healthy adults which consume a high-fat diet and high-sugar diet, a lot of alcohol and medication might have disruptions in the gut barrier. So it is interesting to explore in what conditions prebiotics may interact directly with immune cells, and what is the evidence around the direct effects on immunity. because at the moment only few studies focus on the direct effect of oligosaccharides on immunity. So in order to interact with immune cells, prebiotics need to leak through or be transported across the gut. And here I'm presenting two in vitro studies that provide evidence for intestinal transportation of prebiotics. So in both cases, we can see that prebiotics such as human milk oligosaccharides and short-chain GOS and FOS were able to pass across the gut barrier. In this case, they used an in vitro model of epithelial cells called the CACO2 in vitro model. And the rate of transfer of this prebiotic in vitro was around 10%. These results were supported by human studies in infants. So these infants were fed with formula milk supplemented with prebiotics, including FOS, HMOs, or GOS. And in all cases, these prebiotics were found in plasma, urine, and stools of infants, depending on the degree of polymerization, so the length of those prebiotics. However, although there is convincing evidence to say that some prebiotics can pass through the gut in infants, there is a lack of literature on healthy adults and in those with increased gut permeability. In order to understand whether GOS and also other prebiotics have any direct effect on immunity, 13 different studies were reviewed, including HMOs, FOS, inulin, and GOS. And all prebiotics were shown to directly modulate cytokine production, including some interleukins such as IL-6, IL-8, IL-10, IL-12, MCP1, MIP3 alpha, and TNF alpha, as well as influencing immune cell maturation of lymphocytes and dendritic cells in vitro. And the mechanisms involved appear to be related to ligation to toll-like receptors. And toll-like receptors are membrane proteins that are able to recognize structures from bacteria and elicit an immune response. And only one in vivo study conducted in germ-free mice reinforced this in vitro evidence. So overall, prebiotics can have a direct effect on immunity, which is independent from the microbiota. However, the evidence is different based on the type of prebiotics. For example, HMOs showed a clear anti-inflammatory effect in vitro, which might explain their protective effects against allergy and infection in vivo. However, FOS, inulin, and GOS showed various outcomes, sometimes contrasting, including anti-inflammatory and as well as pro-inflammatory effects. And this may be due to the different doses and types of prebiotic used, as well as different cell culture models and different chain lengths. So because of the role of restoring gut homeostasis and their effects on immunity, there is an increase in interest in using prebiotics as a preventive or support therapy in IBD. So what are IBDs? IBDs are inflammatory bowel diseases, including Crohn's disease and ulcerative colitis. And those are chronic and relapsing conditions that affect the gastrointestinal tract. And although the causes are still unclear, it looks like they are associated with both environmental and genetical causes, and they are associated with inflammation, dysbiosis, which is a condition that refers to alterations in the healthy, normal microbiome, and also increased gut permeability. Unfortunately, there is no current cure for IBD, but there are only maintenance treatments such as use of immunosuppressants, anti-inflammatory drugs, and antibiotics. So what is the level of evidence for use of prebiotics in IBDs? In terms of animal studies, it was shown that various prebiotics, including lactulose, inulin, goat milk oligosaccharides, GOS, and FOS, have a potential in attenuating inflammation, for example, by reducing colonic damage and reducing the amount of secreted pro-inflammatory cytokines. However, more studies are needed in order to use the same experimental colitis models, for example, the DSS model. In terms of human clinical trials, the evidence is a bit weaker. So there are studies supporting evidence for inulin use in IBD. So inulin appears to reduce IBD symptoms and inflammation. However, there are only a few studies with FOS and no studies with GOS. Interestingly, though, when FOS and GOS were used together with other fibers, they were able to increase the levels of IL-10, which is known as an anti-inflammatory cytokine, and also promotes differentiation of Tregs. Overall, however, more research is needed to use more standardized methods and understand the mechanisms involved in prebiotic use in IBD. And I like to finish by mentioning mucosal-associated invariant T cells or MATE cells, because I think they might be a very interesting link between gut and immunity. So mucosal-associated invariant T cells are a subset of T cells which have a key role in immune surveillance, and they are found in high numbers at mucosal surfaces, for example, in the gut, in the lungs, and in the liver. And alterations in their numbers and activation status are often correlated with diseases, including inflammatory bowel diseases. MATE cells are activated by ligands that are produced by gut bacteria during the synthesis of vitamin B or riboflavin. So it will be very interesting to do more studies on prebiotics, probiotics, and understanding their role and if they affect in any way mucosal-associated MATE cells, because literature has shown that unknown metabolites synthesized by probiotic strains are able to activate or modulate MATE cell function. So to finish, I would like just to summarize what I said earlier. So from the literature, we've seen that different types and structures of prebiotics have different effects on immunity. So we saw both anti-inflammatory and pro-inflammatory effects. And there is a lack of studies on transportation of non-digestible oligosaccharides across the gut barrier in healthy and diseased adults. So the current research focuses only on infants. And finally, there is a bit of convincing preliminary data supporting the use of prebiotics as immunomodulators in the management of IBD. However, the mechanism of action are still unclear, so larger standardized studies are needed. So thank you very much for the attention, and I will be here for the Q&A session.
Sorry, everyone, hang on. We're just experiencing a minor technical difficulty. Nathan, if we could check your sound for you.
Can you hear me better now? I apologize.
We can hear you now. Thanks.
Thank you so much, Tracy. I apologize to everyone. The issues of presenting live strike again. Thank you so much, Stefania, for your really, really insightful presentation. I really, really enjoyed it. And I'm sure lots of people have got lots of questions. I know I do. I'd like to ask for now if you're able to just pop off screen again, and we will come back to you in a little while with the open Q&A for everyone. So if you do have questions for Stefania, I'm sure lots of people do, please do start to type them into the Q&A, and we will hopefully get to those in about 10 minutes now. For now, though, I'm going to take over myself, and I'm going to be doing a very quick presentation on the clinical evidence for immune benefits of prebiotics. So following on from these three really great talks that we've already had, my goal really is going to be to provide you all with a really rapid fire overview of where the clinical evidence currently sits for different prebiotics and immune endpoints. I'm going to keep the bulk of what I present as findings from human studies, and there's obviously going to be a major focus here on randomized controlled data as well. before I fully begin, I just want to make it very clear that what I'm presenting to you is just a very small snapshot of some of the published studies currently available and recently published. So it's by no means a comprehensive or systematic review. There are lots of other studies available, I'm sure, lots of other areas where we know trials are currently running and being planned or being published in the process at the moment. So what I've done is selected results from about 20 different clinical trials, systematic reviews, meta-analysis, and that sort of thing. I've only got 10 minutes, so it's going to be really, really rapid fire. It's going to be quite a fun journey for us all, I'm sure. So please forgive me if we do fly through or skip over a few things. I've kept the information as short and sweet as possible, and these slides are available to download, much like all of the other slides as well. All the references are there, so please do use those in the handouts and to find further information. So let's dig into some science. We'll be looking mostly at human data from a variety of endpoints. We're looking at studies that report on how specific prebiotics impact circulating levels of immune cells and biomarkers. Some of the examples we've already heard about a little bit. We're going to be looking at things like immunoglobulins like IgA, T cells, natural killer cells, and other cytokines like interleukins, TNF-alpha, all that sort of thing. We'll also be looking at some real world outcomes like incidence of infection, severity of infection, and that sort of thing. So this is a couple of slides from a very recently published review. We know that prebiotic fibers are conventionally known to serve as a substrate for commensals. We've already seen and discussed many of the pathways that I've got up here, but I thought it's a nice place to start. And that's to just show that many of these metabolites and things like short chain fatty acids can act on various pathways. And in addition to, obviously, as Stefania just presented, having direct effects as well. So there's I wanted to show this because it's a new study. The references there. I think it's really interesting and a great overview. But let's move on to some trial data. And the first set of studies that we're going to look at relate to the effects of prebiotics on specific levels of immune biomarkers and shifts to the microbiome environment. Let's start with a look at GOS and immune functions. This data comes from a 2008 study looking at the influence of GOS in the elderly. It was a double-blind, placebo-controlled crossover study. 44 elderly people were randomly assigned to receive either placebo or GOS at 5.5 grams a day. And the authors found that GOS was significantly increased the number of beneficial bacteria, especially bifidobacteria, at the expense of less beneficial groups compared to baseline or placebos. They also reported significant increases in phagocytosis, natural killer activity, the production of anti-inflammatory cytokines like IL-10, and significant reduction in the production of pro-inflammatory cytokines like IL-6, interleukin-1 beta, and TNF-alpha. So as a result, that team concluded that GOS administration to healthy elderly people resulted in the positive effects on both microbiome, of both microflora composition and immune responses. We can see here some of that data with the placebo on top and the GOS below, and I'll just hold that up for now. We also see some further data from another 2015 follow-up study. This is using the same BGOS ingredient, again at a 5.5 gram per day dose, although I am pretty sure that that 5.5 gram dose is done with a powder that's, I think, only 50 or 60% GOS. So the actual level of GOS in there is actually a little bit lower than 5.5 grams. It's probably closer around 2.73 or something like that. And that found that consumption drove important changes in bacterial populations in the gut, and again had an impact on immune biomarkers like IL-10, IL-8, natural killer cells, C-reactive protein, and all sorts of other great things. I'm going to move on to resistant starches data. This is some data from a very recent meta-analysis on resistant starches, and that reported some really cool results relating to immune biomarkers as well. First of all, you can see here pooled analysis from 7 clinical trials that reported a significant effect on TNF-alpha. And a similar analysis of trials also found an effect for resistant starch on serum IL-6 as well. Another recent meta-analysis looked at pooled effects of prebiotics on metabolic and inflammatory biomarkers. This was a review, a review of 27 studies in total. 9 of them showed an improvement in inflammatory markers, and these benefits were frequently linked to resistant starch, resistant dextrin, oligofructoses, and enriched inulins. The team behind that meta-analysis concluded that there is fair evidence that prebiotics or substrates with prebiotic properties improve metabolic and inflammatory biomarkers associated with type 2 diabetes. This is a 2019 study looking at the effect of dietary prebiotics on inflammatory biomarkers and microbiome shifts in people with colorectal cancer who were going in for operations. We can see in that graph on the right-hand side there how the stress of surgery impacted the abundance of microbial diversity and the gut microbiome environment there. So what's very telling is you can see that how prebiotic intake helped to maintain basically a better relative abundance and microbial diversity there after surgery. The study also reported changes that were linked to improved immune biomarkers, for example, IgG, IgA, CD19, and many others. Now, for clarity, this treatment intervention was a study on a daily dose of 30 grams of prebiotic, and that contained an equal split of FOS, ZOS, polydextrose, and resistant dextrin for a 7-day intervention. Now, this study is a 2019 paper looking at the effect of bacteriophages as a potential prebiotic, and that showed that IL-4 was significantly reduced from baseline. And finally, we have some data on from mouse data showing that human milk oligosaccharides, the 2FL, combined with GOS and FOS boosted flu vaccine responses. And I'll just hold that up for a second as well. I told you that this was going to be a rapid fire thing. So let's move on to health-related primary endpoints. So after that whistle-stop tour of immune biomarkers, let's dig into some real human data. This slide and the next present data from a 2019 review on GI infections and diarrhea in both infants and adults. The table on the right there, or on the left, I think, maybe as you're seeing it, shows some interesting findings, including reduction of upper respiratory tract infections, lower incidence of infectious morbidity, lower intestinal and respiratory infections, reduced infectious episodes, and protection against allergic symptoms. Some of those really interesting things are highlighted in red there. That was in infants. This is the table for adults. There are fewer studies on adults compared to the infants, but we do have a couple of interesting findings here. For example, FOS been shown to be effective at related to C. diff associated diarrhea, but FOS was not alone or sufficient to prevent it. Also, BGOS displayed some noteworthy effects in preventing the incidence and symptoms associated with travelers' diarrhea. And so this is a look at that study, because travelers' diarrhea is actually quite a common condition, especially if you're driving to developing countries, and it's obviously linked to infections with specific bacteria and viruses. So in this study, 2010 study, that GOS was found to reduce travelers' diarrhea by 39% as a real world outcome. So that's pretty great reductions there. This study here gave GOS to university students undergoing exams, which we know is a very stressful time that's linked to all sorts of GI disorders and increased incidence of things like cold and flu. Self-reported stress was positively related to GI symptoms and cold and flu symptom intensity, and the percentage of cold and flu days. The study showed that GOS supplementation modulated all of these symptoms, and a dose of 2.5 grams was actually more effective than a dose of 5 grams at reducing these symptoms, and also had lower abdominal pain. We can see here some data on atopic dermatitis. I won't go into details on this because I am running very short on time already, but you can see that some really interesting findings for GOS-FOS combination relating to atopic dermatitis there. Similar with a 2014 review looking at infectious disease. Data from RCTs there showed that the preventative use of prebiotics decreased the rate of infections requiring antibiotic therapy in infants aged 0 to 24 months, and data from one trial in particular showed that prebiotics may also be effective in decreasing the rate of overall infections. This is just, I wanted to show this quick table very quickly as well. This is just looking at some of that data. And I'm going to flick on very quickly to findings from a 2013 Cochrane review that published a fair few years ago, maybe needs updating, but that found that prebiotics added to infant formula was linked to the prevention of eczema and asthma in infants. Again, another interesting immune-related endpoint. So let's bring this to a interesting but quite controversial contemporary subject, COVID-19 ahead. I do want to start with a very clear warning that there are no clinical trials or other specific evidence linking prebiotic ingredients to COVID outcomes, as far as we are aware, and we're not making any direct claims related to COVID, but what it is interesting to do from a scientific point of view is to look at some of the things that we do know and some of the evidence relating to the microbiome and COVID. So, for example, we know that healthier people with stronger immune responses generally have better COVID outcomes. We know that there is evidence that prebiotic can improve immune responses, as I've demonstrated in some of those slides, and we've seen in the previous 3 talks. Prebiotics also can potentially aid vaccine responses. We've seen some early flu vaccine data there, and also prebiotics can help maintain microbial diversity and abundance in times of medical stress, as shown in that cancer study and many others. And this is, of course, linked to better immune responses and better clinical outcomes. So I just want to show one final study, a couple of charts from that, this 2 more slides really. And this is showing that new evidence has shown associations between gut microbial composition, including levels of cytokines and inflammatory markers in patients with COVID-19. So this suggests that the gut microbiome is involved in the severity and magnitude of COVID-19, possibly via our host immune responses. You can see here the little circles that have got sort of mild, severe and very severe and critical COVID cases and how they separate out. And you can see the track of all sorts of interesting immune data there. These findings also from this study also showed that depletion of immunomodulatory gut microorganisms could be contributing to the severity of that. I'm sorry, I've just flicked the wrong way. This comes with all of the caveats that various other speakers already showed, but this data suggested that the microbiome composition was significantly altered in patients with COVID-19 compared with non-COVID patients. In particular, several gut commensals were with known immunomodulatory potential, circled there, were underrepresented in people who had COVID and remained low in samples collected up to 30 days after disease resolution. So I'm going to end here with another very quick warning that there are no clinical trials linking prebiotics or indeed any other microbiome ingredients, as far as I'm aware, with specific COVID outcomes. We are not making any claims relating to prebiotics and COVID outcomes, and we recommend that you don't either. But it is interesting to talk about in a scientific context, and of course we can't talk about immunity in the current climate without at least mentioning it once. So hopefully that's covered some of the bases. I'm going to stop sharing screens now. We've got maybe 10-15 minutes to do some Q&A sessions now, so I'd like to invite all of these speakers back on. I apologize for my really whistle-stop tour of that science. As I say, all of these questions are, all of the presentations from myself, Pieter, Stephen, and Stefania are available for download, and of course this session is available on demand as well early next week. If Stephen is available to join us back on camera as well, thank you so much, everyone. Welcome back.
Excellent. Shall we just go through this list of Q&A or?
Yeah, so I've got some questions here, and I'm gonna put them to all of you guys. So, I'm gonna come to you actually first, Stephen, if that's okay. And we've had a question in here asking about even with some of the health benefit evidence that we've seen is quite weak with some of the relation to good immune markers. Are you seeing a new structure, function, language appearing in that context?
Maybe. I don't actually know how to answer that.
Yeah.
So
Sure.
structure function claims, you're usually trying to show that it helps maintain some normal process. And sometimes structure function is tricky because at least in the US, the FDA isn't reviewing them. So you don't know if you're actually making a good claim or not until you get in trouble. I think if you can, I don't think the biomarkers are going to be a great way to go about that. We had been asked by clients to look into short chains, for example, how that related to their health benefit. And by the end of this big review, we went, well, there's kind of equal evidence for you and against you. So it's just not a well-researched area. So just stick closer to the health benefit. Of course, the mechanisms provide substantiation, but you have to have a huge amount of data behind your biomarker to
Sure.
really link it to that benefit.
Mm-hmm. So I'm going to come to Pieter now, if that's okay. And everyone, please do feel free to chip in and have a chat here. I'm going to pitch these questions at one person, but I'd love it if other people do come in and throw in their opinion. So this one I'll throw out to Pieter first, but open up to others. Do different prebiotics require different assay methods, Pieter? And if so, how do you differentiate? What are the basic differences there?
Mm-hmm. Yeah, no, indeed. So it's a good question. So it's driven by two directions, I think. So first of all, indeed, you have the composition of the prebiotic substrate as such. So, for example, when you have a very pure ingredient like 2FL-FOS, you can just directly bring the prebiotic of interest in the colon incubation. If, for example, you have a preparation that contains impurities, you might need to do some pre-digestion and also dialysis step. For example, if the compound contains glucose as an impurity, you can just simply do a dialysis with a low kilodalton membrane. If it contains a protein, you really need to digest with pepsin, trypsin, chymotrypsin, apply a different dialysis membrane. And like that, you can then run the sample through the standard colonic incubation setup, let's say. The other question is, of course, the research question, the endpoints that you're interested in. And then it can also really be, you can really do a targeted experiment, because the rule of thumb that I always apply also is to try to keep the experiment as simple as possible. And it's in vitro research that we are doing. So you should not overcomplicate it, because then you're just making it seem as if it's an in vivo test, which it's not the goal of in vitro research. You want to understand mechanisms. You want to do it in a very focused way. So that's would be the answer from my side.
Thanks so much, Pieter. I don't know if anyone has further comments on that, but if not, I'm gonna throw out a question to Stefania as well. Get you involved, if that's okay. We've had a question indirectly to you on whether you see differences between digestible and soluble fibers, if there's similar evidence in different areas there. If you can just elaborate on some of that, that'd be absolutely great.
Yeah. So I'm not an expert on soluble fibers, which are like pectins, gums, which are basically soluble in water. And I know about their health-related effects, for example, in lowering cholesterol and helping controlling glucose levels. But I'm not sure about their effects on immunity, to be honest.
Okay. Thank you so much. So I don't know if you can maybe, I'll throw this out to you first as well, Stefania, but I imagine everyone else will want to chip in on this, and that's whether you can elaborate on some of the roles of short chain fatty acids on immunity and some of the pathways that are involved there.
Yeah. So, well, I think short chain fatty acids affect immunity in several ways. So first, there are receptors for free fatty acids, and so immunity can be affected this way. Also, they are known to lower the luminal pH which also affects immunity because it inhibits the pathogens and also increase the levels of mucin. So it affects the adhesion of pathogens, for example, to the gut epithelium. And another effect is that short-chain fatty acids inhibit the histone deacetylase. So this could be another mechanism through which they affect immunity. For example, they can induce T-rex differentiation through these mechanisms.
Thanks, Stefania. I'm going to come to you, Stephen, as well. I think you've unmuted and look like you want to talk.
Well, yeah, I think Stefania has hit on the head. There's a lot of mechanistic pathways. I was going to say, like a lesson from livestock, for example. We just worked on a paper. So in livestock, they would add short chains, different types, directly to feed, explicitly to reduce incidence of things like salmonella. So formic acid, butyrate has been tested, propionate. And so beyond any of those kind of biochemical mechanistic pathways, short chains, just organic acids in general, have bacteriostatic, bactericidal effects. They will, I mean, this is me saying add it directly to food, so not quite as a prebiotic, but in lowering that liminal pH, as Stefania mentioned, it does create a less hospitable environment for the pathogenic bacteria. So there's very direct effects of short chains, in addition to kind of these secondary signaling pathways that they might elicit.
Great, thank you, Stephen. I don't know if Peter's got anything to add on that topic of short chain fatty acids, and sort of from an assay point of view or an in vitro point of view, whether there's specific things that people should be considering or not.
Oh no, I think mechanistically it was already well elaborated.
Yep.
I think what we just see in vitro, if we have samples with prebiotics that result in large fatty acid production, if you put those samples on immune cells, PBMCs, THP1s, or epithelial cells, you always have a guaranteed nice outcome, let's say. So it's very clear that there's across a lot of different projects that these fatty acids are very potent modulators. So that's very clear.
Great, thank you. I'm gonna keep with you, Peter, actually, but again, I'd like all of our experts to think about this one a little bit, and it's a question about clinical biomarkers relating to prebiotic interventions and where we see the best ones currently. Is it things like IL-6, C-reactive, things like that, or are there other emerging biomarkers that you think could be the most interesting to look at? Certainly, we've covered a lot of different biomarkers today, so I'd love to hear everyone's different opinions on where we see that and whether we see the future going, maybe.
Yeah, so I'm really more focused on in vitro research. So but from my perspective, if I look to the research, clinical trials and biomarkers, when you look at the microbial component of it, it's in vivo. It's a very complex story, of course, because it's a combination of direct effects, indirect effects. The prebiotics they change the pH of the environment. For example, they stimulate mucin production. There's more mucins. They also affect the transit time. Transit time affects the Bristol stool score. There's a nice publication of enterotypes being linked to the Bristol stool score. So if a prebiotic affects the Bristol stool score, it can potentially even change the enterotype of one person across 1 week. So looking at biomarkers on the microbial side, it's tough, but of course, when you control the study very well, it's definitely possible. And there have been many nice studies that you see with novel pectins that are the specialist degraders that have all the mechanisms, like some bacteria species increase nicely. The bifidobacteria that can chew the side chains, they also increase. So there are some nice things to be found. For the immune part, I will probably better leave it to Stephen and Stefania to comment.
Sure, and I'm gonna pitch straight to Stefania, I think, first, if that's okay.
I was thinking of passing it to Stephen. That's okay.
Oh, okay, it's passing me. All right. If you don't already know what biomarker to look at, that means you have to go search for it, which is a painful process and takes forever. It's not painful, it could be fun. In which case, if you don't already have a biomarker in mind, I would say, what is the health benefit you think you're interested in? If it, and usually we do know a lot about different conditions, different immune conditions. We might not know how prebiotics affect them. So I would work backwards. What is the health benefit of interest? Do we know anything about cytokines, interleukins, whatever it is? And maybe start with those. What I also see is if you still can't do that, it's like the cast a super bright net. People are doing XYZ omic assays to try and just get really broad views, gene arrays, things like that. But I would say, is it heretical to say you can sort of skip omic stuff if you already know what the biomarker is, and just be focused that way?
I don't know, is it?
Probably a little bit.
I think it's quite interesting. One of the points you raised, actually, Stephen, in your presentation about sort of almost that middle bit, especially when you're, if you're really looking at a health benefit, like a clinical outcome, like reduced disease severity or something like that, and lower incidence of upper respiratory tract infections, things like that. It's a legitimate question to ask. Does it really matter? All of this in the middle?
I mean, I'm only saying that from a regulatory perspective because sometimes, a regulatory agency isn't going to connect the dots themselves that one thing leads to another. So you want to paint them as clear and direct a picture as possible. If they already have opinions on what a health outcome or a biomarker is, go with that, because it's going to be really hard to convince them that your beautifully designed study, but probably not very well replicated from a bunch of other stuff, does a good job. And it's problematic. Like the newer methods are not as well substantiated because they're new. They were just made, but they're also probably the best. So you're in this catch-22 between like, do I use this older, really well validated method, or do I kind of go with this newer approach? And I don't know the answer to that actually myself.
Thanks, Stephen. Okay, I'm going to ask to wrap up now. I've got one very quick question for Pieter, and I'm afraid we're going to need just a 30-second answer, if that's okay, Pieter. And then I'm going to go through everyone else and just ask if you can again provide maybe 30-second overview. If people only remember one or two things from your presentation and what we've talked about today, what would that be? Pieter, I'm going to come to you first, and just we've had a question here directed towards you. Maybe other people will chip in their 30 seconds. But asking about whether it's possible to study the direct effects of prebiotics in vivo in humans, and how that relates to some of the in vitro things that you're doing as well.
Mm-hmm. Yeah, so I think for the direct effects, an in vivo model is not optimal because there is so many variability. So I think a direct effect of a prebiotic, it's the best possible way to study it is in an in vitro setup where you can really control and avoid the impact of all the other points. And that's also brings me to what I want people to remember from the presentation, is that in vitro models should really very well maintain the in vivo microbial community composition. And the models that I'm developing, they really show that it works much better. So the in vitro model should not result in an adaptation of the in vivo sample to a lab adapted community. It's really about maintaining the human sample as good as possible in the lab, and then to do studies with those in high throughput, address the inter-individual differences between people and not just ignore them by pooling all the samples. And that's for me some of the most important things when you want to really understand what a prebiotic does on the microbiota.
Thanks so much, Pieter there. So I'm going to come to Stefania very quickly, just 30 seconds if you can, on your highlights, and then we'll come to Stephen to round out.
Yeah, I'm happy to add something about studying the direct effects of prebiotics on immunity. So as Pieter said, it's easier to do it in vitro because it would be very nice to be able to do it in vivo, but unfortunately, it is impossible to conduct those studies in humans, for example, because you can do that in germ-free mice models, where you take away the microbiome and you study the direct effects of prebiotics in immunity. But obviously, for ethical reasons, you cannot do that in humans. So yeah, just to add this.
Thanks, Stefania. And Stephen, finally to you.
Takeaways: 1, perform a lit review. 2, figure out what regulatory agencies say about your thing, if anything. And then 3, no matter what study you're doing, I'm of the opinion that you should power it as high as possible instead of going wide with tons of different outcomes.
Great points. Thank you so much, everyone. Really, really fascinating. I'd love to continue the Q&A. We've still got lots of questions to get through, but unfortunately, we have run out of time for part 1, and we have to move on to part 2.